Well, thank you. It's wonderful to be here today, and I wanted to thank Valerie for the invitation. Today, I'm here to tell you about our efforts at UCSF to develop personalized and closed-loop brain stimulation to treat depression. So I have no disclosures, and I'll discuss our clinical trial focused on the goal of personalized neurostimulation and our initial results from our first patient today. Well, first of all, depression is, as many are aware, a very common condition with a prevalence of about 300 million worldwide. It's one of the leading causes of disabilities. And while many people can be managed well with behavioral therapy and pharmacotherapy, there's a cohort of people who are resistant to all current standards of care. And novel technologies such as cortical or subcortical brain stimulation are a potential solution for this group of people. Intracranial brain stimulation, such as deep brain stimulation, has shown promise as a treatment for refractory depression. In this treatment, as shown in the figure, a pulse generator is surgically placed in the chest or skull and delivers a stimulation through implanted electrodes to areas of the brain. So it typically targets a single structure across all people with largely fixed and constant stimulation dosing. And it's been very successful in patients with Parkinson's disease and epilepsy. And deep brain stimulation emerged for depression in 2003, led by the pioneering work of Helen Mayberg. And in open label trials, patients with even the most severe depression achieved remission. And there was a great deal of excitement because it was one of the first treatments that targeted specific brain circuits. But unfortunately, results were not consistently replicated across 3 randomized controlled trials. And the reason may be due to study design. And this figure shows one of the trials, stimulation in the ventral capsule, where the efficacy was assessed at 16 weeks, a time point which maybe in retrospect was too early. However, individual response heterogeneity has likely also played a role. So novel strategies in intracranial brain stimulation treatment are really needed. Personalization of therapy is proposed as a means to improve outcome in medicine generally. The variable presentation of symptoms across individuals with depression and evidence that different neural circuits shown here may underlie different subsets of symptoms really speaks to the need for personalization of therapy and psychiatry as well. And this is an image from Leanne Williams' work. And she highlights here specific circuits underlying symptoms of depression and suggests that knowing the neural circuits involved for each patient might help inform treatment decisions. And we argue that personalization is also needed in brain stimulation. So in depression, multiple anatomical stimulation targets, as shown here in this list, have been identified and have shown promise in small studies. And these areas include the well-known areas, the subgenual cingulate and the ventral capsule, but also other regions in the reward circuitry and newer targets such as the orbital frontal cortex. So it suggests that brain stimulation personalization may be possible. So personalization of therapy includes not only where you stimulate, but also when you stimulate. And ideally, treatment would not be delivered all the time, which could lead to increased side effects or adaptation, but instead delivered only when the symptom severity is high. And that's the idea behind closed loop neurostimulation. So in closed loop neurostimulation, a patient's own physiological activity is used to selectively trigger stimulation when a pathologic state is detected. And there are a couple of devices now on the market that have this capability to continuously sense neural activity. And I'm showing in this figure here the FDA approved and commercially available NeuroPace responsive neurostimulation system, and this is the one that we will be using. However, a symptom-specific biomarker is a precondition for closed-loop therapy, and a biomarker of depression symptom severity has not previously been identified in depression and haven't been identified in most other neuropsychiatric conditions. So direct neural recordings and electrical stimulation have been used in the pre-surgical evaluation of patients with epilepsy in order to identify each individual's seizure focus and map out important functions that are needed to be spared in the surgeries, such as language or motor control. So in this procedure, electrodes are left in place for about a week while patients remain in an inpatient setting. So we thought that a mapping stage could also enable personalization for other neuropsychiatric disorders. And we have trials at UCSF for depression, pain, and obsessive compulsive disorder. And the idea is to extensively map cortical limbic circuitry at the individual level and find a biomarker of high symptom severity state that could be implemented into a closed loop treatment with one of the chronic devices. The ideas behind this goal were developed from years of research through a DARPA subnets program led by Eddie Chang at UCSF. In this research, emotion circuitry was mapped out in patients with epilepsy to identify novel stimulation targets, such as the orbital frontal cortex. And biomarkers of anxiety were identified, and group level biotypes, depression biotypes shown here, were investigated in this epilepsy population, suggesting feasibility. So building on this work, we designed a clinical trial to test the potential efficacy and safety of personalized closed-loop neurostimulation for major depression. The goal was to perform this mapping stage in patients with primary depression and then implement a closed-loop neurostimulation system using the NeuroPace, with the NeuroPace RNS system. And this is an image from our electrode locations from this mapping stage in our first patient. So the trial is called the Presidio trial. In the first stage, we implant 10 16 contact electrodes to map out personalized stimulation and sensing targets and begin to identify a neural biomarker for each patient's symptoms. In the second stage, this biomarker is integrated into closed loop therapy with the NeuroPace RNS system. And in the third stage, we perform a randomized controlled study of initial efficacy and feasibility. Our study population focuses on patients with treatment-resistant depression. A major inclusion criteria to note that are different from other studies are first, evidence that neural circuitry can be modulated, as evidenced by a response to ECT, but not a sustained response. And two, we require that there's sufficient variability in self-reported symptoms to enable biomarker detection. And we send home an iPad with them over a couple of days so they can record their scores. So here's the overall study trial design. So in the first stage of the trial, again, is the implantation of the 10 temporary 16 contact electrodes across 10 brain locations within the cortical limbic circuitry bilaterally. Over the course of the 10 days, the patient stays in the hospital or laboratory setting where we conduct stimulus response mapping to identify the target where stimulation best improves depression symptom severity for the patient. We also pair resting state neural activity with clinical depression scales to identify spectral biomarkers of a high symptom severity burden. If we're able to find a brain site where stimulation leads to a consistent change in clinical symptoms, only then do participants meet criteria to move on to stage 2. And then in stage 2, the NeuroPace device is implanted with 2 4-contact leads placed in locations identified in the first stage. We then have up to a year to validate individual biomarkers and identify stimulation parameters that both modulate depression severity and shift the biomarker back to a healthy state. So once subjects have received a 50% improvement of depression based on a very common depression rating scale, the Montgomery-Åsberg Depression Rating Scale, or 1 year has passed, they qualify to move on to stage 3. And stage 3 is a core study period looking at the short-term and long-term efficacy of the treatment. It follows a crossover design with an intervening 6-month treatment period. And the primary outcome measure, again, is the change in MADRS score across the first and second 6-week crossover periods. We've now completed stage 1 and 2 in a pilot patient. The patient's depression actually went into remission at the end of the stage 2. And we have 3 other participants lined up to start once COVID climate improves. So in the next set of slides, I'll discuss the results from this first pilot patient. So here I'm showing the results of the 10-day clinical mapping study. So during this time, we performed a bipolar stimulation survey across the electrodes in the ventral capsule, subgenual cingulate, hippocampus, amygdala, and orbital frontal cortex. And we saw really a diversity of very specific responses across different sites and different stimulation parameters with just 90 seconds of stimulation. So some examples of this listed out here include tingles of pleasure with 100 Hertz ventral capsule stimulation, neutral alertness, less cobwebs and cotton with 100 Hertz subgenual cortex stimulation, and calm pleasure described as like reading a good book with 1 Hertz OFC stimulation. And then we further explored the brain sites and stimulation parameters that elicit a positive response with sham-controlled stimulation. So we assessed the effect of stimulation using a couple of different clinical measures. So we used a 5-point Likert scale that combines both the subjective response from the patient and physician-rated affect. We used visual analog scales of depression, anxiety, and energy. And we used a 6-question subscale of the commonly used Hamilton Depression Rating Scale that's been used previously to assess short-term changes in depression severity. So results from these scales suggested that stimulation largely modified the arousal and mood valence dimensions in this particular patient. And so we found 3 stimulation paradigms that all reliably improved mood, but were associated with different levels of activation. And that can be shown here in this mood row. You can see the increased mood, positive change in mood in blue, and having a different effect on the arousal dimension. 2 of these paradigms, 100 Hertz stimulation in the subgenual cingulate and ventral capsule, were consistent with prior DBS studies. And the third was a novel location and stimulation condition, low frequency 1 Hertz stimulation across a broad region of the right orbital frontal cortex. So the responses to stimulation at the sites were consistent across trials. Ventral capsule and subgenual cingulate were consistently activating, and the OFC was consistently sedating, as shown in this first figure. However, the effect they had on moods was state dependent. So for example, and shown in this first, in B here, in the OFC, the effect of stimulation was positive and calming if delivered during a high neutral arousal state. But it worsened mood if delivered during a low arousal state, causing the patient to feel excessively drowsy. And the opposite pattern was observed for the subgenual cingulate and ventral capsule regions where stimulation increased arousal. So the effects of stimulation are state dependent. And this patient's primary symptom was anhedonia, and she perceived the most consistent benefit from stimulation in the ventral capsule in areas that's in the reward pathway. We also found that the response to stimulation was dose dependent, shown here for the ventral capsule on both mood and arousal dimensions. And we found that the response to stimulation was sustained beyond the stimulation period. The stim period is shown here in black, and you can see the effects extend further, even up to 40 minutes after ventral capsule stimulation. So these results, both the dose dependence, the persistence of the effect, and the state dependence, suggest to us that closed loop stimulation may be a feasible treatment option and was worth further exploration. So in addition to clinical mapping, we also did biomarker exploration during this period. So we had patients perform 335 VAS scales of depression and anxiety over the 10 days, which we used to define two symptom severity states shown here with blue and pink, a low symptom severity state and a high symptom severity state. We then identified 15 minutes of resting state intracranial EEG activity that coincided with the clinical measures where one of the two states was present. And we made sure that those 15 minutes were away from any stimulation as well. We then identified the intracranial EEG spectral activity features that discriminated the high and low symptom severity states using six standard frequency bands and two cross-validated supervised machine learning models at different spatial resolutions. So in the first model shown at the top, spectral power was averaged across all contacts within brain regions. And we found that gamma power in the bilateral amygdala, right OFC, left subgenual cingulate, and right hippocampus had high state discriminative potential with an accuracy of 76%. In the second model shown below, the feature set was defined by spectral power for each electrode, which allowed us to account for interregional variability due to the electrode positioning. So in this model, we found that amygdala gamma power alone was sufficient for detecting the high symptom severity state with an accuracy of 77%. So together, the findings suggested that amygdala gamma power is a biomarker in this patient of a highly depressed state. So the mapping stage for the purpose of personalized target selection and biomarker development was the first to our knowledge in a psychiatric disorder such as depression. We characterized the complex interplay between symptoms, mood state, and neural stimulation, and we demonstrated the importance of a patient's symptom profile in interpreting the clinical response to stimulation. So these results suggest that the time a patient spends in a particular mood state could be a consideration in the selection of a stimulation target. We began stage 2 for this patient on June 1st of last year. We implanted the NeuroPace device with two separate sensing and stimulation depth leads based on the mapping study. The RNS system neurostimulator, shown here, continuously senses neural activity, and detection of pre-specified patterns of activity are coupled to delivery of electrical stimulation to the designated electrodes. And we use DTI in the planning of the surgery so that the contacts interface with two fiber tracks that we found in the first stage, the stria terminalis and the ansa peduncularis. We then assessed whether we could replicate the clinical effect of the VCVS stimulation that we saw in the mapping study. And we found that at the expected contacts based on anatomy, we again saw acute symptom improvement across the majority of trials where the patient, across the majority of trials shown here in purple. The patient described the effect again as pleasurable and energizing. And we saw the opposite effect with stimulation in the amygdala shown in green. It made her symptoms worse. So in traditional stimulation, you modify some parameters, including frequency, pulse width, and current amplitude. But in closed loop stimulation, we have to consider another parameter, burst duration. So to do so, we tested the clinical effects of intermittent stimulation at different stimulation burst durations over 15 and 30 minutes. And we found that just 6 seconds of intermittent stimulation at 1 milliamp was clinically effective, and importantly was below this patient's perceptual threshold. So that means that the on-off states of the stimulation were not detectable to the patient and therefore would not likely interfere with her normal functioning. And that's what's shown in this figure. We also found that we were able to replicate the symptom-specific biomarker for closed-loop control. So we used the NeuroPace RNS system telemetry wand to stream wireless activity in the laboratory. So the patient actually sat in a TMS chair, and we held it with a TMS flexible metal arm over her head and over the neurostimulator. We recorded 10-minute trials of continuous amygdala activity while the patient engaged in naturalistic activities such as using social media or talking to her family on the phone, watching movies, or even sometimes performed her virtual therapy appointment while we streamed the data. We then paired the neural activity with clinical ratings. And again, we found that amygdala gamma power, shown here, was highly correlated with both the VAS depression rating scale and the HAM-D6, the Hamilton Depression Rating Scale. Furthermore, it significantly differentiated the high symptom severity state from the low symptom severity state. So in summary, we replicated the effects of the mapping study with the NeuroPace device. The effect of the clinical response to VCVS stimulation was positive and energizing, and the amygdala gamma power was again found to be predictive of symptom severity state. So we're currently determining whether we can implement this biomarker into closed loop control. But the initial results are encouraging for us. The first patient, in this first patient, her depression is currently in remission and she's doing quite well. But we have a lot of work left to do. We don't yet know how variable these findings will be across individuals and whether the short-term responses that we're seeing now will lead to long-term treatment of major depression. The results do suggest, however, that quantitative circuit-based treatment development is possible in depression. And this approach is just one of a number of exciting advancements in personalization of brain stimulation and complements the work of others, including Helen Mayberg and Samir Sheth, who are also working on similar goals. So the framework also is not unique to depression, but could be applied to any circuit-based disorder, so a broad range of neuropsychiatric conditions. And indeed, in UCSF, we have studies underway in OCD, chronic pain, and mood disorders of Parkinson's disease. So the work I presented is made possible due to a broad collaborative multidisciplinary team, including Edward Chang, who conceptualized much of this work, Phil Starr, Ankit Kambadi, Ghassan Makul, and Andrew Crystal, who leads the broader brain stimulation program in the psychiatry department, and collaborators Vikram Rao in neurology and Leo Sugru in neuroradiology. And of course, our funders, NIMH, NINDS, the 1907 Trailblazer Award, and the Ray and Dagmar Dolby Family Fund. So thank you, and I'm happy to take questions.